Monoclonal antibodies are typically produced in mammalian cell cultures and afterwards purified through a series of unit operations. The purification process is called the downstream process. Here we see a lab-scale setup of an integrated continuous downstream process for monoclonal antibody purification. The setup includes 2 chromatography systems performing the purification steps. The process, which is fully integrated and automated, and requires minimal operator intervention, is started via the computers controlling the chromatography equipment. This is our starting material, a clarified mammalian cell culture containing our product. To achieve a product of high purity, host cell proteins, viruses, DNA, and aggregated antibody need to be removed. The first step is the capture step, where the antibody binds to protein A with high affinity. Here, 3 parallel columns are operated in a periodic counter current setup. The multi-column setup enables a continuous inflow of clarified cell culture to the process, and leads to increased productivity and reduced buffer consumption. The capture step is followed by virus inactivation. The product eluted from a capture column is pooled in a stirred flask, and held at low pH for around 1 hour. Further purification is performed through 2 polishing steps, based on ion exchange and mixed-mode chromatography. Finally, the purified product is collected. To control and automate the process, a Python-based software called Orbit is used. With Orbit, customized flow paths and advanced pooling strategies are easy to implement. The downstream process requires several buffers. Normally, these are prepared by hand, but in this setup, a buffer management system automatically supplies all needed buffers. The buffer management system monitors the buffer volumes and prepares fresh buffer when needed. Buffer is prepared according to predefined recipes by mixing stock solutions with water. After the buffer has been prepared, it is transferred to the correct buffer flask. The operators can easily make adjustments in the Orbit software, if necessary. The presented integrated continuous downstream process for monoclonal antibody purification, including the buffer management system, is fully automated, and the process can run without any operators present.